Hello and welcome. In this video, we will be discussing the AutoFIND F, an innovative automated nanotechnology for application in fish by Tethys. Fish is a well-established method since the 80s, still performed today with minor improvements, requiring trained technologists and skilled manual execution, which can have significant variance from technologist to technologist depending on the skill level of the technologist. The recent increase in request for fish analysis has significantly raised laboratory cost and boosted technological advances for automation and rationalization of the assay. Fish Standard Protocol is composed by a series of steps, each of them more or less relevant according to the specific cell sample in analysis. Here we show the general method with manual slide preparation, aging and sample pretreatment, up to probe addition, and slide sealing with rubber cement for hybridization. Among these steps, some of them are critical and can depend on uncontrolled variables of the external environment, such as temperature and humidity, or technical skills. The aim of Tethys is to bring fish to efficiency through automation, by simplifying and rationalizing the technologist's work of setting up and processing samples, to reduce the time needed to perform the analysis so that the technologist can be dedicated to qualifying activities. Tethys developed a system that automates the fish procedure from sample dropping through hybridization. AutoFIND F is a liquid handling platform that performs interphase and metaphase fish on cytogenetic samples. The majority of fish steps, from sample loading to hybridization, have been automated. AutoFIND F executes fish protocol on a proprietary slide called MicroFIND. Tethys has developed MicroFIND, which is a slide with a proprietary coating coupled with microfluidic compartments or microchannels, where cells are automatically loaded and processed for fish. After hybridization, slides are further analyzed by fluorescence microscopy using conventional or automated imaging systems. MicroFIND slides are designed to provide the processing of up to 4 hybridizations within the microchannels. The essential feature of the MicroFIND is the nanocoating, which functions to collect and immobilize cells in a spatially confined area of a microchannel where hybridization can take place. In this video, a cytogenetic pellet is loaded on MicroFIND and in parallel on a commercially coated slide coupled with the same microchannel device, showing the superior capability of the nanocoating to efficiently retain cells in shear stress condition, allowing fish to be performed. AutoFIND F automatically dispenses samples which form a monolayer of cells that stay stable until the end of the fish process. According to cell type and sample features, cell numbers can vary between as few as 100 up to 4,000 cells. Suitable samples for use within the AutoFIND F for fish are cytogenetic pellets and carnoise fixative from blood, bone marrow, and urine samples. Cytogenetic pellet preparation is achieved according to conventional methods through manual or automated harvesting. Other suitable samples are living cells coming from peripheral blood after red cell lysis, or samples collected with affinity isolation methods, like bone marrow processed using column or magnet plasma isolation. Here are some of the AutoFIND F features. Samples and reagent racks can be easily removed and placed back on the machine. Working probe volume is reduced down to 0.7 microliters per hybridization, and sample volume is reduced down to 2 microliters per hybridization. Reagent consumption and waste production are significantly minimized. AutoFIND F offers fully automated or semi-automated modes for probe loading for the highest probe savings. It executes different protocols in parallel according to sample features and according to specific customer needs. A unique protocol allows the utilization of living cells coming directly from red blood cell lysis or cell separation through column or magnet whenever the making of cytogenetic pellet is hampered or difficult due to low sample availability. Fixation is performed inside the channel. Using 2D barcodes, traceability is accomplished by associating patient to slide and probe to microchannel positions on the work table. Work table and reagents are verified before each run using our dedicated conductive tips. AutoFIND F is equipped with disposable tips to prevent any cross-contamination, removable probe and sample tubes, removable reagent tubes, independently thermostatic microfind units, webcam for barcode scanning, a complete automated humidification system, and a computer with an integrated software system. Here is an overview of the operations performed by the technologist to run the AutoFIND F. Clicking on the Find Cube icon, the software is launched. Different tabs will show up, allowing you to select number and type of protocols. You can run 4 different protocols in parallel, depending on the nature of the sample and code denaturation parameters. After selection of the protocol types and code denaturation parameters, the operator will enter sample IDs and associated probes. Labeled tubes are filled with up to 10 milliliters of reagents. Warning windows remind the operator to fill these reagents and to check system water and waste levels. Right after reagent loading, microfinds are labeled, assembled, and placed in position according to the software indication on the work table. The dedicated assembling tool provides precise and consistent assembling of the slides. Positioning of the slides is visually helped by the software. Now, probes and samples are loaded into the dedicated vials. Each vial holder is then loaded into the AutoFIND F, which is now ready to start the execution of the selected protocols. First, the work table check is performed and samples are dispensed, along with pretreatment reagents, probes, then mineral oil for microchannel sealing. Finally, microfind thermostatic units are closed for hybridization. Once hybridization has ended, microchannels are removed using the dedicated disassembling tool. Then the technologists can proceed with their usual post-hybridization wash following the instructions provided by the probe manufacturer. Once the microfind slides have been washed, they can be mounted with an appropriate counterstain and imaged using a standard fluorescence microscope or an automated imaging system. Here are the advantages of the AutoFIND technology. Protocol standardization. Reliable, high-quality data generated by standardizing FISH protocol. Reduction in reagents requirement. Up to a 14-fold reduction of reagents volume significantly reduces the cost of each test. Easy image acquisition. Narrow and reproducible scanning areas reduce image acquisition time in both manual and automatic scanning platforms. Lab productivity improvement. Automation frees up time for the technologist to work on qualifying tasks. About 1 hour of technologist time is required for 64 hybridizations. Thank you, and please visit our website for further information.